Our first speaker and our second speaker today is Dr. Michelli Oliveira. She is a postdoctoral fellow working with Davy Smith and Sarah Gianella. She's been there since 2013. She earned her PhD at the Federal University of Rio de Janeiro, and her interests include the dynamics of HIV populations in body compartments and neuropathogenesis of HIV. And so her first talk, it will be entitled Compartmentalized HIV DNA Populations Persist in CSF Despite Suppressive ART. Oh, you're good. All right.
All right. So the title of this study is Compartmentalized HIV DNA Populations Persist in Cerebrospinal Fluid Despite Suppressive Antiretroviral Therapy. After transmission, HIV can fastly disseminate through the different body compartments. And as an example, in the case of the central nervous system, studies have been shown that HIV RNA can be actually detected very shortly after infection, and studies have been shown as early as 2 weeks after transmission. So generally, in these body compartments, HIV is able to establish reservoirs that can persist over the course of infection despite long-term suppressive antiretroviral therapy. During the course of infection, HIV also may suffer different evolutionary pressures inside these different anatomic tissues and also adapt to them, leading to what we call virus compartmentalization, which is an indicative of local replication in these different tissues. In the central nervous system, it's thought that resident macrophages and microglial cells, they may serve as the main source of HIV replication in this compartment. And perhaps they may also serve as HIV latent reservoir in these tissues. However, little is known about the characteristics of these HIV DNA compartments, especially during suppressive antiretroviral therapy, and specifically, what would be the effect of early initiation of ART in these compartments in terms of the characteristics of these HIV DNA reservoirs. So, the question that led to this study was if early initiation of antiretroviral therapy would be associated with lower levels of HIV DNA and inflammation in the cerebrospinal fluid and the CNS compartmentalization status compared to initiating ART later during the course of infection. So for this study, we prospectively collect paired blood and 40 ml of cerebrospinal fluid samples from 16 HIV-infected individuals that were enrolled from the San Diego primary infection cohort. And they had continuous suppressive levels of HIV RNA. 9 of these subjects start antiretroviral therapy within 4 months after the estimated date of infection, and 7 of them start antiretroviral therapy later than 14 months from EDI. In this table, we have the summary of the baseline clinical and demographic characteristics for this cohort. And mostly this cohort was composed by Caucasian males with a median of age of 45 years, with good CD4 counts, and they present the median of 2.6 years of lifetime exposure to antiretroviral therapy. And none of these characteristics was significantly different between the two ART initiation groups. We measure in all these individuals soluble markers of immune activation, CD163, IL-6, TNF-alpha, MCP1 in blood and CSF, and marker of neuronal damage measured by the neurofilament chain light, or NfL, in CSF. We also measure the levels of cell-associated HIV DNA by droplet digital PCR in PBMC and CSF cell pellets. We finally generated ultra-deep sequencing in the region of envelope from HIV that were amplified from genomic DNA from blood and CSF cell pellets using the Roche 454 platform. And we use this sequence for the study of compartmentalization between blood and CSF. So first we look at difference for the levels of inflammation between the two ART initiation groups. And we observed overall that levels of inflammation as shown by the levels of IL-6 and TNF-alpha in cerebrospinal fluid was associated with early initiation of antiretroviral therapy. We didn't find difference for levels of the other markers of inflammation, neurofilament chain, and also we didn't see difference between the two groups for neurocognitive impairment as measured by global deficit score. We detected HIV DNA in most of samples from PBMC and around 62% of the CSF cell pellet samples. And we haven't found difference regarding detectability or levels of HIV DNA in PBMC and CSF when compared to the two groups. We were able to amplify and sequence samples from PBMC from 14 subjects and from CSF cell pellets from 90 subjects of this cohort. And although we haven't seen a difference regarding percentage of diversity from sequences from PBMC, we observed a trend towards early initiation of ART presenting lower percentage of diversity in envelope. About the studies of compartmentalization, we were able to sequence paired PBMC and CSF cell pellets from 8 subjects in this cohort. And here is a table summarizing the compartmentalization analysis. And we saw that all patients but one presented significant signal of compartmentalization between CSF and blood, and including the 3 subjects that start ART during acute infection. So we also generated phylogenetic trees using the sequence obtained from PBMC and CSF from all the patients. And here we have represented by the red circles sequence from blood and the blue diamond sequence obtained from CSF cells. And we also saw that in the majority of the patients, we were able to detect CSF-only sequences that were not present in blood. And again, even when ART was initiated during acute infection. We were also able to amplify and sequence from 2 individuals longitudinal time point, where we can see again CSF-only sequences that were present in the very first time point, and that persist over time as we could detect in the second time point. So as conclusions, HIV DNA in CSF was detectable in more than half of HIV-infected subjects, despite suppressive antiretroviral therapy, including the subjects that start antiretroviral therapy during early HIV infection. Early initiation of ART was associated with lower levels of inflammation, as shown by levels of IL-6 and TNF-alpha, but no difference was observed for HIV DNA levels and neurocognitive impairment related to early initiation of ART in this limited cohort. And despite of ART initiation, we saw that a compartmentalized HIV DNA reservoir may establish shortly after infection. And one of the limitations of our study is obviously the sample size, and we believe that cohorts including a larger number of samples and longitudinal time points can help us to confirm our results and also help us to understand the dynamics of HIV reservoirs in the central nervous system. I would like to thank you, the study participants of this study, also all the collaborators for this work that collaborated directly or indirectly, and also I'd like to thank you for your attention.
Sequence-only clones. I mean, do you think it was just kind of a founder effect that, you know, infected cells that cross the barrier and then, you know, or do you think there's something else going on as far as immune pressure?
Well, we don't actually know, and this is what we're trying to look at. So this is still very debated for all the studies. So it could be a founder effect, but also it can be a sign of local replication in this compartment. And this is important when you think about strategies of eradication, because if it's true that that HIV can establish a latent reservoir in the CNS, so we have to look at it in the CNS when we talk about strategies of eradication.
Did you look for RNA in the CSF?
No, we looked, all these patients, they were totally suppressed during all this time. And we actually measure at the level of single copy of RNA. And all of patients, they were undetectable, totally undetectable.
So how do you explain the differences that you've seen in your inflammatory markers if you didn't see a difference in DNA and you have undetectable RNA?
That's a good question. I cannot explain based on this data.